Hey, and welcome to the next installment of my E-Biology Teacher Podcast. I'm Mr. Doc, and today we're talking about enzymes. Yes, those pesky little enzymes. Well, that might not be very fair. Enzymes are incredibly important. As a matter of fact, one could say that enzymes are responsible for you being alive right now. So you better learn about them, and you better appreciate your enzymes like your grandma told you to. Grandma might not have told you to appreciate your enzymes, but if she knew about enzymes, she probably would have told you to be appreciative of your enzymes. All right, so we are going to talk about enzymes today. We've already talked about the different major molecules in organic chemistry: carbohydrates, proteins, lipids, and nucleic acids. Enzymes are a type of protein, but they deserve their whole show by themselves. Okay, they're that important. So hang tight as we talk about how enzymes function. Why they are important to living organisms in general, what substrates they interact with and how they do that, and also talk about what happens if the enzymes that we do use stop working properly. All right? So before we get started too much, there's one thing that you've got to understand in biology but also everywhere else, and that is form determines function, meaning the shape of something determines what it does. Very simple. Now, if the shape of something changes, one of two things are going to happen. Either the function is going to change, or sounds like I just cursed there. Or beep. No. Or it's just going to stop working. So it's either going to stop working or it's going to take on a new function. Now, if you take a look at this fork, yes, it's a fork, or it once was a fork. It would not be very good for what a fork is normally used for. That is because the shape has changed. Now, it would be hard to even get food on here, much less be able to put it in your mouth. All right? And then you're more likely to put out an eye than get food in your mouth, right? That's because the shape of the fork has changed, right? So shape is incredibly important. When we're talking about tools, when we're talking about biology, we're talking about chemistry, we're talking about physics, we're talking about anything that has an action, shape becomes very, very important. Form determines function. If we understand that, then everything else becomes a little bit easier. All right? Now, the next thing we need to talk about is reaction rates. Now, a rate, and this is a good little thing to learn in no matter what science you're talking about, a rate is any measured change over time. So if we're talking about our rate of speed, that's the distance we're traveling over the amount of time it took us to travel that distance. If we're talking about let's say consumption rate of electricity, it's kilowatts per hour. If we're talking about how fast we're burning calories, it's calories per hour, calories per minute. If you're talking about how fast somebody's running, it's meters per second. Those are all rates. And in biochemistry and in enzymes, we start talking about reaction rates, which is obviously how long it takes a reaction to occur. Now, there are some body processes that simply take too long to happen. So your body uses proteins called enzymes to speed up the reaction rate. In other words, we make these reactions happen faster, which is what a catalyst does. And after all, enzymes are just biological catalysts that increase the speed at which a reaction will occur. Now, I do want to take a quick note here and say that enzymes can't make a reaction happen that wouldn't naturally happen anyway. It can just make it happen quicker, okay? So if we were to put a whole cheeseburger right in our stomach without any enzymes, it would take weeks, if not months, to digest. It wouldn't just fall apart. But because we have enzymes, it speeds that process up. So instead of a matter of weeks and hours, or excuse me, weeks and months, we're talking about a process of hours. Very, very quickly things are going on. And that's what enzymes really accomplish for us, is that they allow us to do processes that would really take just an unfathomable amount of time. There are some processes that have to happen that quick, otherwise we can't survive. So that's why enzymes are so incredibly important, because it allows those chemical reactions that have to happen in our body to happen quick enough to keep us alive. Okay, and that's obviously incredibly important. So none of the reactions that enzymes do just wouldn't happen naturally, but many, very, very many of the reactions wouldn't happen fast enough, and that's why enzymes are incredibly, incredibly important. That means that you can find enzymes pretty much anywhere. In the digestive tract, in DNA replication, protein synthesis, filtering blood. And enzymes are both catabolic and anabolic depending on the enzyme itself. Catabolic means that they break down things, and anabolic means that they build up things. The word anabolic is probably familiar to you from anabolic steroids, and anabolic steroids are taken by people to, what? Build up more muscle. So that makes complete and total sense. There is sort of a misnomer or a lack of understanding about enzymes, because people will often say, oh, enzymes help break things down faster. That's both true and misleading, because while they do help in some cases break things down, they can also help by building certain things up. Okay, so I don't know that we want to restrict it to just breaking things down, because not all enzymes break down. Some are anabolic in nature, meaning that they can actually help us build things as opposed to tearing things down. All right, but they are located pretty much everywhere, and that makes them pretty important. Enzymes work in a very simple way in terms of what we call the induced fit model. The induced fit model says that an enzyme has a place on it called the active site, and a substrate, or the substance that that enzyme is going to work with, has a complementary shape that fits into the active site. Once the substrate has fit into the active site, the enzyme can either connect the substrates or break the substrates down or do whatever it wants to. In other words, it fits like a lock to a key. And this is an example of an enzyme. You'll notice that the substrate has this shape, and that the active site has the complementary shape, meaning that this is whole, and then the hole in the enzyme is actually empty, which is the same size as the substrate, the same shape. So that would fit in. Now, this is usually an animation, and for some reason I can't get it to launch right now, but if this were to enter into the active site here, the enzyme would rip it apart, and we would be left with these two pieces of red thing. Those would be my products. Okay? Now, the other thing I want you to note is this. While the substrate is broken down, the enzyme is not changed at all. That means that enzymes can be reused. Enzymes are not reactants. They're not products. They are just accelerants. They go in, they make the process happen quicker, and then they can be hopped out and they can go again. So that enzyme that you just saw on your screen, not only can it break down that substrate, but once that substrate is turned into products, it can take another substrate and do the same thing over and over and over and over again. All right, so so enzymes aren't used up in that process. Now, before we go on any further, there is a little analogy that I like to use when I'm talking about enzymes. This lock represents a strand of DNA. All right, now if I try to open my DNA, it's not going to happen very easily. I can keep pulling and pulling, and maybe eventually I could break the lock, but I very seriously doubt that. Now, what I can do is use a whole bunch of different enzymes. See my enzymes? Yeah, they're keys, I get it, but it's an analogy. Work with me. All right, so we've got all these enzymes here, and what I start doing is I start using the enzymes, and then I realize that this enzyme doesn't work, and this enzyme doesn't really work. But this enzyme, now that works, okay? Because the enzyme has to match the shape of the substrate. So that means that these little teeth on the key fit on the substrate, and that's why I could unlock this particular molecule. Now, if I were to try to take this enzyme, which looks a lot like this enzyme, but you can see that it's broken, it doesn't do the trick. So if I change the shape of the enzyme, then I change its function, meaning that this key will no longer work. The same is true with just a little change. Let's say, for example, that this is a lipid, and I have its enzyme right here. Pop, right? Now, I have the same enzyme here, but I want you to look at something. You can barely see it, but there's that little bend right there. Watch what happens. It won't fit. And if it doesn't fit, you must have quit. Kidding. That's a O.J. Simpson joke and a really bad one. Sorry. All right. So that one doesn't fit because it's been changed. So that's a pretty good lesson for us. What happens when proteins change shape? Well, enzymes can change shape. And there are many factors that can do it. If an enzyme changes shape, then it's not going to work properly because remember, it's all about shape and fitting. If an enzyme changes shape, then it won't work anymore, or it will take on a new function. And things that can change its shape include heat or pH. Viruses and mutations can change shape, and there are plenty of other factors that do that. This protein on your screen is unfolded right here, but this protein is folded, meaning that this is the exact same chemical compound, except it's taken on a different shape. Whether you know it or not, you've actually seen denaturing happening before. Frying an egg. An egg has a lot of protein in it, but when you crack it into that pan, it's all clear and liquidy, it hits that hot pan and instantly turns white and starts to convert to a solid. That's because the proteins in the egg are changing shape. They are loose and goosey until you put heat on them and then they fold up, which is why you end up with that solid egg. And because they dense up, they also change color and they change their state. So they're not quite liquidy, they're actually pretty solid. That is the denaturing of a protein. Well, the same is true of an enzyme. An enzyme is a protein. If the enzyme changes shape, then the active site isn't the same. If the active site and the substrate don't match, there is no enzyme action, and we don't get to have any fun. Sad. All right. So, the last thing I want to speak about is the specificity of enzymes. What I mean by that is that enzymes are specific to the substrate that they use. The active site of an enzyme has a specific shape, the substrate has a specific shape, and those shapes must fit like a lock to a key. In other words, they must be complementary. This means that enzymes only work with a specific substrate. That means that for every chemical reaction you have in your body that requires an enzyme, it requires a very own enzyme. It can't use another enzyme. I can't have an enzyme that breaks down carbs, or let's say an enzyme that breaks down DNA. It can't be the same enzyme that breaks down lipids, because lipids and DNA have completely different shapes, and therefore the enzymes that break down DNA wouldn't fit for the lipids. So they have to have different enzymes. That looks kind of funny. Boom, 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 boom. All right. So that's our story about specificity. Enzymes only work for the things that they match, which means you have thousands of chemical reactions happening in your body by the use of enzymes, which means you have thousands of different enzymes in your body to do those thousands of different chemical processes. All right? Now, I'm going to show you this quick video, and hopefully this will give you an idea of how enzymes work in your digestive system. Hold on one second, here we go.
Food reaches the end of the esophagus, it goes into the stomach. Inside the stomach, both mechanical and chemical digestion take place. Mechanical digestion occurs as the stomach squeezes the food, churning it and turning it into a kind of soupy mixture. Chemical digestion occurs when glands that line the walls of the stomach secrete gastric juice. Gastric juice contains hydrochloric acid and several enzymes. The hydrochloric acid kills germs in the stomach and helps the different enzymes work. One of these enzymes is pepsin. Pepsin breaks down molecules of protein into simpler molecules. In small children, another enzyme, renin, helps digest a protein found in milk. The enzymes and hydrochloric acid in gastric juice are so strong that they could eat away the stomach wall itself. However, the walls of the stomach are coated with a lining of mucus that protects the stomach from the chemical.
All right, we get the idea, I believe. Now, there's something that may have popped into your head while you were watching that going, wow, there's hydrochloric acid in my stomach and there are enzymes. And Mr. Doc said that if the pH is too high, then the enzymes will change shape, which means they won't work. That can't happen. Well, it's all about being in the right place at the right time. You see, enzymes, just like any other chemical reaction, have a optimal condition, okay? Meaning that they'll happen best at a certain temperature and a certain pH and all that other stuff. The enzymes that are in your stomach work better at a lower pH, meaning more acidic. Whereas the enzymes, say, in the nucleus of your cells that make DNA, they prefer a neutral environment, okay? Everything has a optimal range, a narrow range of temperature and pH and concentration where this is where I'm happiest. If we move out of there, that doesn't mean that the equation can't happen. It just means it's not going to happen as well. An example of that might be, let's say you start your car on a very, very cold winter morning. The car kind of, I don't want to start, it's too cold. And then all of a sudden it gets warmed up, and once it gets warmed up, it just moves fine down the road and you're good to go. Well, in the summertime that doesn't happen because the processes, the chemical processes that make your car go, happen best between that range of, say, 50 degrees and 80 degrees. All right. It also explains why a car will overheat in the summer, because it's just too hot for those chemical reactions to happen more efficiently. It's also why our cars don't run as efficiently in the heat of the summertime. So just like a car has an optimal range of temperatures to operate in, doesn't mean the car won't work in the winter. It just has to work harder to make it happen. The same is true of enzymes. Now, if the stomach enzymes are exposed to too much acidity or too little acidity, that's when they'll change shape. And when they change shape, of course, they no longer work. Let's take a look at our final objectives and see what we learned. First of all, describe the enzymes' importance to living organisms. Well, we know that enzymes help reactions happen at a fast enough rate to keep us surviving. If we had to wait for those reactions to happen on their own, they would happen too slow, and we would die and die and die a painful death. Not to put too fine a point on it. Enzymes only work with a specific substrate because it's all about shape. If the substrate doesn't match the active site, then that enzyme simply will not work. Enzymes work because the substrate attaches to the active site because of their complementary shape, and the enzyme is either able to put together or break apart that molecule based on what is needed. And finally, if an enzyme changes shape, it means that either one, it will just not work, or number two, it will take on a different function. I hope this information has helped you understand and appreciate enzymes, and I hope that you take the time to appreciate enzymes as well, because it is enzymes, after all, that make you alive, because otherwise everything would happen so slow that you would not be here, which means you would not be watching this, which means that you would not be learning, which means I've pretty much wasted my time. So thank you, enzymes, for all that you do for us. I'll see you next time on your eBiology Teacher Podcast.